All right, welcome back, everybody. So we're continuing our journey with Chapter 7, our Energy and Thermodynamics and Enzymes chapter. This last little bit before we jump into enzymes is we're going to take a look at some special reactions and energy transfer, primarily ATP hydrolysis and redox reactions. So first of all, we're going to take a look. At ATP. So we've kind of thrown this term around a couple times already. You may have seen it in your book, but ATP stands for adenosine triphosphate. So here is the full name of it. And basically what adenosine triphosphate is we have this nitrogenous base molecule, very similar to the A in DNA, like the A's, T's, C's, and G's, the adenine base of a nucleotide, one of the nucleotides. So we have adenine, which we can see as carbon, nitrogen, and hydrogen. It is connected to a ribose sugar, so this is a monosaccharide, it's a 5-carbon sugar. And then if you take a look at it, we have 3 phosphate groups. Remember, phosphate groups were one of our functional groups back in chapter 3, kind of gives particular properties to organic molecules. So when you have adenosine triphosphate, you have those 3 components. You have the base, you have the sugar, and you have the phosphate groups. The reason why ATP is so valuable to our cells, it is a really good energy storage molecule. And the key bond that stores a lot of that potential energy is this guy right here. So it is that bond between the 3rd phosphate group and the 2nd phosphate group. So when ATP is formed, it usually starts as adenosine diphosphate. So we have the adenine, we have the ribose, and we have 2 sugars. That's the di. And then we squeeze on one of these 3rd phosphate groups. So in the process of going from ADP, the diphosphate, to ATP, the triphosphate, is we have to cram. Now, if you take a look, let me erase some of these markings up here to make it a little bit more clear. So if we take a look at the charges on these phosphate groups, we have a negative charges on these oxygens, right? Now, what do we know about like charges? Do they like to attract or do they repel? Okay. Opposite charges attract, like charges repel. So if you have these 2 really negatively charged phosphate groups and you're trying to cram on another negatively charged phosphate group, that's not going to be super easy. So you have to have some kind of tool, which is going to be an enzyme, which we haven't talked about those yet, they're coming up next, and an input of energy to be able to cram that 3rd phosphate group onto those original 2 phosphate groups. And by doing that, what that's going to do is it's going to make this bond right here a really high energy bond. And so that's where this potential energy of ATP is stored, is in that bond between the 2nd and 3rd phosphates. So when you do a hydrolysis reaction, right, we know hydrolysis means water breaking. So with some enzymes, you add water and then you break apart your diphosphate from what we just call inorganic phosphate. A lot of times you'll just see it P subscript I, just letting you know that's just a phosphate group hanging out all by itself. So in this process, that's going to release a lot of energy. This is a highly exergonic reaction because there's a lot more energy in an ATP molecule and a lot less energy in the ADP molecule. So again, if you do final minus initial, the hydrolysis of ATP has a negative delta G, and we'll see that in this next slide here. So how does the cell take advantage of that potential energy? Or the ability to actually make ATP. So how do we cram that 3rd phosphate group onto the diphosphate? So in this picture over on this side, here we can see, in this box, we can see exergonic reactions. So this is something that's happening in the cell, say like cellular respirations. We're setting that up for chapter 8. If you remember that slide that I did on the last video of my catabolic pathways on one side, we had like our carbohydrates, lipids, proteins catabolized into the energy poor substances, and one of that byproduct off into the middle part of that slide was energy, and ATP was a form of that. So the energy that's stored in our sugar, the food that we eat, and then fats, and proteins. The energy that's stored in the food that we eat, it is released in these catabolic reactions, and that energy is used to drive that cramming of that phosphate group onto the ADP to generate ATP. So we're basically transferring the energy. We know that from our first law of thermodynamics, energy can be transferred. We're not destroying it, we're not making it. We're just transferring it from the chemical bonds that are in your sugar or your fats or your proteins, whatever is being digested. We are transferring that energy from that molecule into an ATP. And our cells just spend ATP better. Sometimes you might hear ATP as being called the currency, energy currency of the cell, just because a lot of our enzymes and the reactions are set up to take the energy from the ATP as opposed directly from a glucose molecule or a lipid or an amino acid. So we have to have some kind of intermediate process to take all of that energy that's stored from the food that we eat that ultimately came from the sun, right? We have to have a way to convert that potential energy in the sugar molecules into our usable form for our cells in the form of ATP. And so that's what's happening in this pathway. It is exergonic reactions in our cell is driving the endergonic, so this is a positive delta G, to turn ADP into ATP. Okay, and that's what chapter 8 is going to cover, is how do we make ATP, that cellular respiration. So now let's say we have some ATP, it's hanging out in our cell, we want to do stuff. Let's build a protein, or let's replicate our DNA, or let's endocytose or exocytose, or let's active transport, anything that the cell needs energy for, we have our bank filled with ATP. So what we'll do is we will hydrolyze ATP, which is a negative delta G reaction. So it's spontaneous, going from a high energy ATP to a lower energy ADP. And that energy given off will drive our endergonic reactions, the metabolic reactions that our cells need to maintain homeostasis. Okay, so it's this cycle. So the food that we eat, we extract the energy out of that food to make ATP. Then we use the ATP to drive cellular processes to maintain homeostasis. So we know if you don't eat food, you die. And one of the reasons on the cellular level why you die is because you're running out of food to drive the production of ATP. And if your cells don't have ATP, they cannot do the processes needed to maintain homeostasis. And if your cells, tissues, organs, organism gets out of too much out of homeostasis, then your body systems are going to start to fail, and that can lead to death. So food is important, but now you know why. Not only because it tastes good and it fills you up, but it is our raw materials, not only to make more of who we are, but to drive this process of energy transfer from the potential energy in the food to the potential energy of ATP, which is the form of energy that our cells can actually use. So let's take a look at some of these reactions that we can use ATP for. We're going to start, okay? So we're just going to start taking a look at the delta Gs. And again, you don't need to memorize these numbers, but it's more of the concept of this idea of what's called reaction coupling. So we can couple the reaction of one exergonic reaction to drive an endergonic reaction. So let's first take a look at the actual values for ATP hydrolysis. So when you go ATP with water, because that's part of the hydrolysis reaction, your result is ADP and an inorganic phosphate plus some hydrogen ions. So if we look at the delta G is a negative 32 kilojoules per mole, which is a unit of energy. We don't need to be super familiar with that. So would the hydrolysis of ATP be considered an endergonic or an exergonic reaction? I'll give you a minute to think about it. We looked at what delta Gs mean in the last video, positive versus negative. So a negative delta G, final minus initial, meaning your final products have less energy than your initial products. So ADP has less energy than ATP. So you're going to get a negative delta G, which is going to be a, drumroll, exergonic reaction. Good. So if you chose exergonic, way to go. Negative delta Gs are always going to be an exergonic reaction. And we just talked about how the hydrolysis of ATP releases energy. We're breaking that bond between the second and third phosphate, and that releases energy. All right, let's take a look at another reaction that you might have in your cell. It is what's called the phosphorylation of glucose. So glucose is a molecule that's often drawn like this, and it's got a carbon up here. So sometimes we hook a phosphate group onto that carbon, so called glucose 6-phosphate. So in the reaction of adding a phosphate group to your carbon molecule, here's our reactants, glucose and phosphate, and here's our product, a glucose phosphate. It's all one molecule now with some water, so the dehydration synthesis. And if we take a look at the delta G is positive 14. So what that means is the glucose 6-phosphate has more energy than the glucose and the phosphate separately. So if the products have more energy than the reactants, is this an exergonic or an endergonic reaction? Right? It's a positive delta G. So endergonic reaction. So it is endergonic because it requires energy to phosphorylate that glucose. So these were two reactions that were kind of calculated separately, the hydrolysis of ATP and the phosphorylation of glucose. Now, what if you were to kind of combine the reactants in these two reactions into one reaction? And so this is the coupling part. So let's say your coupled reaction is the glucose, right? And the ATP. And I'm not showing the water and the hydrogen, but know that they're there, but for ease of, or for clarity's sake. And so then our product is glucose 6-phosphate, and then our ADP and our ADP. So we have all of the same reactants, we have all of the same products. Now, how are we going to figure out if we're to couple the reaction, the hydrolysis of ATP with the phosphorylation of glucose, could we predict whether this is a spontaneous or non-spontaneous reaction? Well, what's kind of nice about knowing those delta G's is all you have to do is add them together. So if you were to add the negative 32 of the hydrolysis of ATP plus the 14 of the phosphorylation of glucose, you sum those together, you get a net delta G of negative 18. So with this coupled reaction, would this be an exergonic or an endergonic reaction? And so the E word that's being blocked off by my head right there is effect. So coupled reaction has a net exergonic or endergonic effect. What do you think? A negative delta G is going to give you a net exergonic effect. So the phosphorylation of glucose will be happening spontaneously when you couple the hydrolysis of ATP and that phosphorylation of glucose, because the hydrolysis of ATP releases so much energy, it has the ability to cover what's needed for this endergonic reaction with extra. So this coupled reaction is going to be spontaneous. All right, so this is a real life example that's happening inside of your cells all of the time, using the free energy, this negative 32 kilojoules per mole of ATP hydrolysis, to drive endergonic reactions. It's happening all the time. And we'll see more of these examples as we dive deeper into chapter 8. Okay, and the last little bit I want to talk about are these redox reactions. So you talked a little bit about it or read a little bit about it in chapter 2. You had one answer on your one of your case studies for redox reactions. So just to review what these mean, redox reactions are also called oxidation reduction reactions. These, I have a little saying here that might help you remember. It says, Leo the lion says grr. So Leo stands for losing electrons is oxidation, and grr stands for gaining electrons is reduction. So anytime electrons get transferred from one place to the next, one thing will be oxidized, that's the one losing the electron, and one thing's going to be reduced, and that's gaining the electron. And I know it can be a little bit hard to wrap your brain around that, because things being reduced are gaining something, and that doesn't compute right away, because you're like, if I'm gaining something, I shouldn't be reduced. But what are they gaining? They're gaining negative charges. These electrons are negative charges. So gaining electrons is going to reduce your overall charge. You're going to become more negative. So that's the reduction part that we're talking about. So keep this saying in mind. Leo the lion says grr. We're going to see a lot of redox reactions in cellular respiration. So just try to keep this in mind. You can always come back and rewatch this section of the video if you need to when we get into chapter 8. So when we are transferring electrons, they typically don't fly off from one atom to the next all by themselves. We saw that with the sodium and chlorine. So if you remember, we had sodium ion, it donated its electron to chlorine, and then they became sodium chloride. That was a direct transfer of the valence electrons. In redox reactions, a lot of times the electron is donated with a hydrogen atom. So you might have like a hydrogen atom plus an extra electron just kind of hanging next to it because the proton is a positively charged nucleus. It's got its original electron and it might have this extra electron that it's going to be carrying with it to add onto another molecule. So just keep that in mind. We'll see it in some examples. So the big picture, right? The big picture idea when we get into chapter 8 is this reaction of glucose, because glucose is the food that we're eating. We're going to just focus on glucose, but know that all sugars kind of get boiled down to this process. Glucose gets oxidized into carbon dioxide, okay? Oxidized, losing electrons. So what we're doing is we are stripping electrons away from glucose. We're stripping in multiple reactions. That's all of cellular respiration, the first 3 stages of cellular respiration. We're stripping electrons away from glucose. And who are they going to? They are going to be going to oxygen. So oxygen is going to be reduced into water, right? So let me change this here. So the electrons that are hanging out in this large organic molecule actually go to oxygen with hydrogen, right? So here's the oxygen, here's the oxygen, here's some of the hydrogens, here's some of the hydrogens. And so your electrons that you're stripping away from the glucose get transferred to the oxygen. And here's our reactants and here's our products. So glucose in the presence of oxygen will be oxidized into carbon dioxide. Oxygen will gain those electrons and be reduced into water. Okay, so we can't do it just in one quick reaction though, because it would burn up. We would turn into charcoal or explode if we're to transfer electrons straight from hydrogen onto oxygen. It just doesn't work that way. So we have to do it in these tiny little steps, and we'll see that when we get into chapter 8. But some of these intermediate players that we will be seeing, I'm going to introduce you to those in these next few slides, are the key players in some of these redox reactions that we're going to be seeing in cellular respiration. So the first one is called NAD and NADH, stands for nicotinamide adenine dinucleotide. I think that kind of sounds like that's what it should be. So we have it in 2 forms and it's reversible. So seeing these reversible arrows tells us that when we are going to add the hydrogen and electron onto NAD+, it will become an NADH. Or we can take those hydrogens and electrons away from NADH and convert that to NAD+. So here are the electrons in the blue. So these are what has been stripped off of something else. So something just got oxidized. And now we are going to reduce NAD+. And so these guys just come and stick right here. And so we can see that right here. And some electrons here. So NADH is the reduced form. It just gained electrons. And we can tell that because it's got a new hydrogen attached to it. And I just said electrons transfer with hydrogen. So whenever you see that hydrogen stuck to the NAD, that lets you know it just received some electrons with hydrogen. If we see no hydrogen, we know that the hydrogen and the electrons, because we see the plus charge there, have been removed. So when NADH gets oxidized, if we're going this way, it's losing electrons to become oxidized, gaining electrons to be reduced. So NADH is an electron carrier. We're going to call it an electron carrier. It has the ability to take up electrons as we're stripping them away from our glucose and temporarily hold on to them until they drop them off somewhere else in what's called the electron transport chain, which we'll be seeing in chapter 8. Another very similar molecule is called FAD, and again, it is reversible to FADH2. So again, here are the electrons and the hydrogens that are being transferred, either gained, so this is the reduced form, or lost, this is the oxidized form. Okay, so FAD and FADH2 again are another temporary electron carrier. As we are stripping electrons away from our glucose, FAD will hold on to them temporarily, becoming FADH, and then they will drop them off at the electron transport chain. So I just wanted to introduce to you these two intermediates, the NADH and the FADH2. We're going to see them again in chapter 8, but now you have hopefully kind of a sense of these redox reactions, where the electrons are going and where they're coming from, as these play a part in the whole big picture of trying to strip the electrons away from glucose, dropping them off onto oxygen, and in the process, we get some ATP. All right, so that is our reactions and energy mini lesson, and then I have one more lesson from chapter 7 talking about enzymes, which are basically the tools that allow all of these chemical reactions to happen. All right, I will see you next time.